formation is achieved through different stages. After cleaning, Ar-Rahman Ar-Rahim. The lecture of today is about the microbiological aspect of dental caries. The relation between dental plaque or biofilm and dental caries is going to be discussed, and also certain hypotheses concerning different types of bacteria and the initiation and progression of dental caries. Following eruption of teeth and exposure to oral environment, bacteria will be able to attach to and adhere to different teeth surfaces, and this aggregation is known as the biofilm or dental plaque. Dental plaque is a diverse microbial community found on tooth surfaces, embedded in a matrix of polymers. These polymers are of bacterial and salivary origin. 80 to 90% of the weight of a plaque is composed of water, and 70% of the dry weight of a plaque is formed from bacteria. The remainder is a matrix of polysaccharide in addition to salivary proteins and glycoproteins. Dental plaque is considered to be the etiological factors of many diseases, especially dental caries, periodontitis, gingivitis, and peri-implantitis. You should remember that the dental plaque or biofilm are present also on healthy teeth. Plaque formation is achieved through different stages. After cleaning of teeth and within a few seconds, glycoprotein originated from saliva are going to be absorbed to the tooth surface, forming what is known as the acquired pellicle. So, the acquired pellicle is the first stage in dental plaque formation. Acquired pellicle is very important in the protection of teeth surfaces, as it's going to protect the tooth from the transfer of ion from the outer enamel surface to the surrounding environment. Within 24 hours, there will be a passive transport of oral bacteria to the tooth surfaces, adhere to the acquired pellicle through a reversible process. This reversible adherence is achieved using weak, long-range physico-chemical interaction between the glycoprotein and the acquired pellicle and the microbial cell surfaces. This type of interaction is through van der Waals attraction and repulsive electrostatic force. The third stage in plaque formation is through the irreversible attachment of what is known as pioneer microbial colonizers. Here, the type of interaction is through specific receptors present in the cell walls of bacteria and complementary receptors, proteins on the pellicle surfaces. The first bacteria to adhere are cocci and other aerobic bacteria, while in pits and fishers and proximal surfaces. The main type of bacteria are Streptococcus, Gram-positive. Within 7 days, the pioneer bacteria will create an environment that attract other types of bacteria, and there will be a gradual replacement of the already existing bacteria to allow for other species to adhere and to grow. The co-aggregation or co-adhesions of the late colonizers are through adhesion receptor interaction. These interactions build up the biofilm to create a more diverse environment, which include the development of unusual morphological structures like corn cobs and rosettes. After 7 days, a climax community or a mature biofilm will form. This includes multiplication and biofilm formation, including the synthesis of extra polysaccharide. The last stage of plaque formation is the detachments of bacteria from surfaces. The microbial composition of dental plaque differs between the supra and subgingival plaque. There is also a diversity in the bacterial number, types, and the proportion from time to time and from side to side within the mouth in the same individual. The plaque composition is very important as it is going to determine the pathogenicity of this plaque. And this pathogenicity differs according to the different types of tooth surfaces. We have two terms. We have primary colonizer bacteria and secondary colonizers. The primary colonizers are the first bacteria that adhere to teeth surfaces. And usually they are in an insufficient number to be pathogenic. And also most of times they are aerobic. That means they are oxygen tolerant. The first bacteria to adhere to the tooth surfaces are the cocci, especially Streptococcus, in addition to actinomycetes, gram-positive facultative rods. As the thickness of a plaque increases, there is a deficiency in oxygen supply, so other types of bacteria going to be attached, and this is called secondary colonizers bacteria. An example of these bacteria, we have the gram-negative cocci. The amount and rate of bacterial composition are affected by different factors like the toothbrushing, sugar consumption, and the oral immune system of saliva. There are evidence from animal studies and human observational and epidemiological study to prove that dental caries is a microbial disease. In order to have dental caries, we should have cariogenic bacteria in dental plaque. This cariogenic bacteria can ferment carbohydrate, and the end result is acid, thus dental caries. So, the presence of microbial communities is considered to be a prerequisite for caries to develop. Let's discuss first evidence from animal studies. From different experimental animal studies, it has been shown that germ-free animals fed cariogenic diet, they develop dental caries only following the transfer of bacteria from another animals. So cariogenic bacteria can be transferred from one to another animals. That's why dental caries is considered to be a transmissible disease. So it is a transmissible and infectious disease. Other studies show that antibiotics and chemotherapeutic agents can arrest active lesion or prevent new lesion. These are proofs that this disease is a microbial disease. Animal studies showed also that there is a specificity of bacteria to develop caries according to type of tooth surfaces. As you can see in this table, the predominant bacteria in smooth carious lesion involve Acimutans, Salivarius for pits and fisher, Acimutans, Acidogenicity, Salivarius, etc., in addition to lactobacilli and actinomyces. While for root caries, the predominant bacteria involve actinomyces and other anaerobic bacteria. For proximal lesions, the predominant bacteria involve actinomyces, gram-negative bacteria, in addition to streptococcus and Acimutans. Evidence from human studies. Epidemiological and observational studies reported an increase in the prevalence and severity of dental caries with the increase in the number of cariogenic bacteria, precisely mutant streptococcus and lactobacilli, in the saliva and dental plaque. In similarity to animal studies, it has been reported a variation in types of bacteria according to tooth sites. And from human studies, it has been found that Acimutans and lactobacilli are the most carcinogenic bacteria compared to other types of bacteria in the oral cavity. Virulence, or the carcinogenic potential, is referred to different bacterial properties that contribute to the caries formation. This involves first acidogenicity. What do we mean by acidogenicity is the ability for production of acid by fermentation of carbohydrate. Acid formation is the prerequisite for cariogenicity. The second cariogenic potential is the acid production rate. The capacity of acid production vary by different types of bacteria. Some types of bacteria produce acid faster than other types. For example, streptococcus produce acid in a faster rate compared to actinomyces. This type of acid, for example, lactic acid, can be produced by metabolism of saccharose by bacteria. Lactic acid can cause demineralization of the outer enamel surface, in another word, dental caries. Another carcinogenic determinant or carcinogenic potential of bacteria should be aciduric. Certain groups of bacteria are more acidic than others. So we can say that these bacteria can tolerate acidity more than other type. Lactobacilli and Streptococcus mutans can grow in a medium of pH 5 and 5.2, thus acidic medium, while other type of bacteria cannot survive in this medium. An important carcinogenic potential of carcinogenic bacteria is their ability to produce an extracellular polysaccharides. For example, glucans, which is an insoluble type of extracellular polysaccharides. Glucans is very important because it is the key for adherence of bacteria to tooth surface and build up of microorganisms on teeth. The extracellular polysaccharide is considered to be a diffusion barrier that prevent the diffusion of acid to the surrounding environment, keeping the acid in contact with the tooth surface, increase the probabilities of dental caries formation. The final virulence factor of carcinogenic bacteria is their ability to form a storage of polysaccharide. These bacteria may have the ability to convert a portion of sugar, if present in excess, to an intracellular storage polysaccharides. So when there is deficiency in oxygen, or deficiency in exogenous carbohydrate, they can utilize and metabolize this intracellular storage for energy. Finally, acid can be formed, and there is more probability to have dental caries. Mutans Streptococcus is considered to be a carcinogenic bacteria, as from different animal and human studies, a positive correlation was reported between the counts of these bacteria with the increased prevalence and severity of dental caries. They possess carcinogenic potential properties, including acidogenicity, aciduricity, and ability to produce extra and intracellular polysaccharide. These bacteria were isolated from surfaces immediately before development of dental caries. So these bacteria are considered to be the more carcinogenic potential bacteria and responsible for initiation of dental caries. Lactobacilli are considered to be carcinogenic also, as a positive correlation was reported between their counts and dental caries in enamel, dentine, and root surfaces. Lactobacilli possess carcinogenic potential as they are acidogenic, aciduric, and able to produce extra and intracellular polysaccharide. But unlike Streptococcus, lactobacilli are rarely isolated from a plaque before development of carious lesion, so it has been suggested that lactobacilli may be responsible for a progression of dental caries and not for initiation of dental caries. vivo and in vitro studies reported an association between actinomyces and root caries. These bacteria were isolated from dental plaque samples taken from root caries lesion. However, other studies are needed to clarify their role in relation to caries development. There are different hypotheses regarding the role of plaque in relation to dental caries. The first hypothesis is known as specific plaque hypothesis. According to this hypothesis, only a few bacterial species are actively involved in the production of dental caries. From mutans streptococci, only 2 subspecies are responsible for dental caries in human teeth. These subspecies are Streptococcus mutans and Streptococcus sopranus. According to this hypothesis, preventive measures should be directed to these subspecies to limit their number, thus control the disease. The other hypothesis, it is known as the non-specific plaque hypothesis, and according to this, dental caries is the outcome of the overall activity of total plaque microflora. So, a heterogeneous mixture of microorganisms are responsible for this disease and not a specific type of bacteria. The third hypothesis is called the ecological plaque hypothesis. According to this hypothesis, dental caries is a consequence of imbalances in the resident microflora resulting from changes in the local surrounding environmental condition. The change in the surrounding environmental condition is caused by acid, and this acid is the end result of fermentation of carbohydrate by different cariogenic bacteria. The general strategies of caries prevention, according to plaque hypothesis, involve first, inhibition of plaque acid production. This can be achieved by using agent, for example, fluoride. Fluoride in high concentration not only can improve the remineralization of the outer enamel surface, but it considered to be toxic to bacteria or able to inhibit certain bacterial enzymes responsible for metabolism of bacteria. By this, we can prevent acid formation, and this acid is important for the survival of cariogenic bacteria. The second point in our strategy is to avoid between meals food and drinks in order to give chance for buffer capacity of saliva to have again a neutral pH, and by this to prevent acidic medium continuous present surrounding the outer enamel surface. Again, the acidity is a good environment for bacteria to survive and metabolize. The third strategy is stimulation of salivary flow after main meals. Stimulation of salivary flow will increase the buffer capacity and also will remove a remnant of carbohydrate and bacteria from the oral cavity. By this, we can promote remineralization and return the pH of a plaque to resting levels. Thanks for your kind attention. And the other slide gonna be a simple question to be answered in the Google class. Answer briefly. Can we remove the supra and subgingival plaque by only rinsing the mouth with water and salt? Explain.